It's a great pleasure to have Jorn here. I mean, he's going to tell us about something which is intriguing, and so which is topological statistics in living and non-living matter.
Yeah, thank you, Tani. Also to the organizers in general, and also for all of you coming. I know there's a strong competition going on, and so in the upper corner, basically, I show the person who should actually give that talk. So Dominic, I'll tell a little bit more about him. You will see him many times. So he did 90% of the work I'll present, both intellectually and computationally. And he's actually from here, so I'll mention it more. Yeah, so what I would like to present here is basically recent work where we were interested in comparing disordered systems. And I will show basically experimental pictures that, provide some motivation. But if you remember Sebastian Streichen's talk, so you were showing these organoids, for example, where people create organoids, but it's very difficult to compare them in any form. So we are curious if how much information you could actually strip away and still be able to compare structures, basically packed structures. But what I wanted to do for the first 10 minutes or so is just also because I'm still here for the week, give a very brief overview of some of the things we're interested in. So if there are questions or some people are interested in discussing, so I would be very happy to talk. Yeah. Okay. I'm scared that it's not okay.
Yeah.
Okay.
That's better, I think. Yeah, try the other one.
Well, it's the front is off now.
Okay, good.
So we are broadly interested in collaborating with experimentalists, basically understanding collective dynamics of living systems. So here's an example Alex Mieke talked about, so I won't talk too much about. It's very similar to the systems that Ray has been studying for a long time. And we're also interested in programming active materials or living materials. So here, basically, let's work with Ingmar Cruz's lab at the University of Arizona, and they're able to genetically engineer bacteria so that they bind basically according to certain rules. And so what you can do is you can seed them here, basically being in these bright spots. And then basically, if you let the system evolve, basically it grows interfaces according to the rules that you can pre-program. So it's basically a design problem there. So one hope that we have is basically by understanding collective dynamics and maybe introducing some controls that we will be able to also produce something eventually that could be useful in application. And here, basically, I will talk or show basically one example of recent work with Saad Bamna's lab at Georgia Tech. They have this very interesting system of filamentous active matter, I would call it. So you could also say simply blackworms. And so what these guys do is when you basically put them in a Petri dish, video plays, and you watch them for several minutes. So then they will build a very dense tangle. And that's basically natural behavior in response to certain basically environmental conditions. So here you can see Saad, basically, who performed all the experiments. So they've been studying that system for a long time before we became involved. The experiments were done by Harry Altuazon and Emily Kaufmann also had, and Vishal, who is also from Cambridge originally. So was a PhD student in my group, he developed some theory. So one of the things that we are interested in, or we're interested in there, is to understand basically both this tangling behavior and also something that's very curious. So if you look at the time scale here, so this is really now several minutes basically until you get this very dense. Was that the things? Now it's restarting my computer. Yeah, so yeah, but it's just as you push the button. Yeah, so it happened also. As a projector went off, it probably felt it lost an arm or so. And
there's some exotic electronic interference between that.
Okay, so the IT guys.
Is
No,
just keep going.
Go and tell us.
Let's see how it works. Good.
Yeah.
Yeah. So what I wanted to point out is basically that it took several minutes to produce this dense tangle. But if you try to gently electrocute these worms, so this is real-time video, you'll see two electrodes coming in. So the system disentangles basically in basically sub-seconds. And so one interesting question was, these organisms are fairly simple, so a very basic muscular system, neural system. So how are they able, basically with very basic physical rules, to accommodate these two behaviors? And this is just basically the slow-mo version of that. And yeah, so it turns out that basically with a very basic mechanical model with a little bit of noise, activity, and friction, basically you can replicate that behavior, and then you can actually make proposals for how to design basically efficient tunneling protocols. Also, if you were interested in building robots of this type, also. Yeah.
It almost looked like the fluid motion from the wires was perhaps as important as the.
Maybe. So we don't know exactly what creates that. So they do that basically on a different stimuli. So it's basically an escape response. But the fact is they can still do it very fast. So it's definitely not that the wire steered them and they flew apart. So it's more there's some.
I'm wondering whether they sense the motion.
It's very well possible. So they're still continuing that, basically starting the system. It's extremely interesting. So once these blobs have formed, they can actually collectively migrate. They can transport things around once they basically find an obstacle. And they can also pull themselves up at the liquid air interface, which is also quite remarkable form, basically like a chimney type thing where, I mean, they don't know really if it's because of biology, so that they want to mix basically or get some oxygen in or so, but it's a very interesting system. And it, and most of the systems I've been working on were typically point-like or rod-like, and these are actually filamentous with particles that do interesting things. Good. So the other questions that we are interested in is also how we can store information in active and soft matter. So I will not talk too much about it. So that's the middle part. But we spend quite, I think probably 50 or 60% of our resources now will be on this part, where we try to build inference pipelines that allow us to translate imaging data into biophysical models, constrained biophysical models, hopefully basically in a quantitative way. So that basically by feeding in a video under sufficiently constrained model, we're able to measure several, say, hydrodynamic coefficients simultaneously, or couplings, force couplings simultaneously, and so on. So just to give you an idea, so how that typically works. So the first step is always trying to come up with some sort of low-dimensional representation. And we borrow here basically knowledge from the description of polymers or other physical systems. So this is basically one system that we used as a test case, which is data that was provided by Jan Huisken's lab. So that was published basically independently before. So this is development of a zebrafish, or first basically 10 hours roughly after fertilization. And you see that the cells start out as a hemisphere, basically, and then eventually basically start to migrate on a surface, basically form basically this elongated structure, and then also will form basically what will be vertebrate in the future. So the way we were basically trying to use that as a test system is to project basically in that case onto suitable basis functions, for example, scalar spherical harmonics for the density fields, vector spherical harmonics for the flow field. And then it turns out that you can actually get away with a quite limited number of modes, basically, to capture this type of dynamics. So instead of writing down a PDE, basically, which is technically infinite dimensional, so you can try to infer finite dimensional ODE systems. And it turns out basically, so here is this one system that we consider. So it's basically a simple thing you can come up with, or you think of that as basically mode vectors and mode amplitudes. And this is your dynamical matrix that creates the dynamics. Then you typically have to satisfy certain constraints. And the question is basically given data for these modes, basically, can we infer something about the dynamics? And then maybe, because I mean, these typically are very simple systems, then use other techniques to classify basically also collective behaviors. In this case, it turns out that this matrix basically has this interesting structure. And it turns out basically that the main structure that you see is actually comes just from active Brownian motion. So you can run an active Brownian motion model on the sphere, similar to what Dunkel has been doing this week. A long time ago. And if you do analytic coarse graining there, so you exactly expect these type of structures. So the interesting part, biologically interesting part, is probably in these off-diagonal elements, which we don't quite understand yet. So this is one example, but the approach is quite generic. So we can use basically whatever basis system may be suitable or symmetry informed or otherwise informed basically for a specific system. So in another recent work, basically used something that is very similar to study undulatory locomotion, basically of snakes, worms, robots, basically various things. And it turns out in that case, because you have a length constraint or an approximate length constraint for many of the things, you can actually map that to Schrödinger equations. I know that Ray basically has done work on similar mappings also a while ago that are very useful. And so what is interesting about this type of basically mapping is that now you can basically use tools from what I would say, quantum theory or just linear systems theory to classify behaviors, because different behaviors are mapped basically onto different matrices. So you can study the eigenspace of these matrices and so on, give you classification. Just to give you an idea, basically how these models do. So this is basically for straight moving states. So you can also do convents. So the data basically here are these experimental data are these circles, and then basically this is what a linear model would give you, basically fitted to the data. And the reason why we're actually interested in that is not so much basically the physical part of the dynamics, but our collaborators, especially in Stephen Flavel's lab, they're able also to simultaneously measure neural dynamics. And so what we would like to do is eventually connect the neural dynamics with the physical dynamics, but we want to ideally do that basically by using low-dimensional representation in both spaces. So that's basically also something we are interested in. Good. So now for today, I would like to focus on a slightly different topic, which I chose because there's statistics in the title of this workshop, and this is the thing that is most statistical, I would say, in our recent work. Yeah, so as I mentioned before, so this work was mainly driven by Dominic Skinner, who is now an NSF fellow, so he has a PhD student in my group. And I think he was one of your top students in part 3. By top, I mean really top. So very good and really amazing person who will soon be on the job market. So if you want somebody who's exceptional, I would encourage you to talk to him. And so Dominic developed basically a lot of the algorithmic ideas. So we'll show you basically there are some algorithmic challenges in comparing these disordered structures. He also developed analytical, basically at some stage also proofs or rigorous results. And Boya Song, another PhD student in my group, provided some test data. And then we obtained basically data from various collaborators, basically that we used to test basically these methods that Dom had developed, and also basically previously published data. And the goal was basically to understand basically how we can characterize basically disorder structures in general, using some ideas that are very common basically in, say, computer graphics, for example, but maybe not so common in physics yet. So in particular, ideas from optimal transport, which probably many of you have heard of, but maybe some of you haven't. Good. So the motivation, as I mentioned, is essentially the revolution in current imaging techniques. So you can get basically high resolution data as single cell, even in some cases dynamical data, basically at single cell resolution. So for bacterial system or for embryonic tissues, this was basically what I showed, basically system, organoids, and some models. Here's another basically plant-based system. I could have shown basically many other systems here. But what is often quite difficult is to compare these systems. What I'm interested in is also, so how could you compare basically across system boundaries? So not just basically understanding one specific system. So you could, for example, ask, if I change the bacteria slightly, so I introduce a mutation. So how would I basically change the structure of the system? But I would also like to understand now how similar at a certain level are, for example, these embryonic tissues to biofilms or something. So what it means is we have to strip away a lot of useful information that we would have to keep if we're interested in a specific system. So, but the thing that you could basically strip away while still keeping some useful information is typically basically, or what you could keep is the topological information. So neighborhood information about how cells are basically connected to each other or which neighborhoods they have. So this is one of the goals. And once we started doing that, so we found something that we still don't quite fully understand. But it turns out that there seems to be generally a difference between how living systems and how non-living systems pack, basically at a topological level. So I show you some purely empirical evidence. But so we found it curious, and I will show at the end basically a web page where Dominic basically has collecting data, and if any of you have data basically from different systems, please, when you want them basically added to that atlas that I'll show at the end, please send them to us. Good. So just very briefly to just explain how topological characterization differs from geometric characterization, which has been successfully used in many different contexts in tissue dynamics and tissue studies in general. So here's an example from Lisa Manning's group. So for example, you can study vertex models where you represent cells basically by areas and interfaces, and you have an energetic penalty. And what you try to understand is how you can make the vertices active or so. So there are various basically knobs to play with. And what you could try to understand is how the behavior of these models changes basically in response to, or can be diagnosed just by looking at geometric quantities. So one parameter that is quite common, or is purely geometrical in a sense, is you compare the average parameter to the square root of the area, you get a dimensionless number. And then basically is a statement in the paper that says basically once you exceed the critical value under certain conditions basically on your model, you get a transition basically from a solid to a fluid behavior. So this is clearly a geometric criterion. So it's not something we are interested in. So we are interested more in topological, so going basically one level, basically coarser. Another very nice example, basically from recent work is by Ray's group with Radcliffe and Peter Janker at Georgia Tech. So where they were looking at volume distributions basically in cell packings, and they found that you can basically very often observe modified gamma distribution that you can actually derive from entropic arguments. So they validated that on basically a multicellular yeast model that was developed at Georgia Tech, and also basically a Volvox model that was studied in Ray's lab. And so that suggests over there's some universality also basically in understanding these type of systems. So what we're interested in is really just something coarser, slightly coarser. So we are interested in just topology, and our work basically, our starting point for our work is previous papers by in particular Emmanuel Lazar, and basically he collaborated for quite some time with Prof. McPherson. And so what they were interested in is basically characterizing two-dimensional, three-dimensional topological packings. So what we build on top, basically using their work, is basically optimal transport ideas to compare. Good. So what you can see here is just a packing basically of points. You think of them as cell nuclei, or colloids, or whatever basically you would like them to be. And so now you can wonder, so why would topology basically be a useful way of characterizing at least some of the information? So if you color now all these points basically by the neighbors, so that's taken from their paper. So you can clearly see basically that there's some structure here. So there are these scars, and then you have basically here a localized defect structure and so on. So that suggests that by looking at topology, you capture at least something. Now,
Jorn, is that just a Voronoi tessellation?
it's just a Voronoi tessellation, very simple basically.
So these cells are generic cells or living cells? Or?
Oh, and I think in their case, they just generated basically one example, but you can do that basically for tissues or everything. So I'll show at the end basically. So that's the goal basically. I'm working towards it. So we try to find as much data as we could and put them into one big, basically topological phase diagram or atlas or whatever you want to call it. So the way you can now basically characterize these topological pattern or structure is by looking at specific neighborhoods, basically of any arbitrarily picked cell here. So here I show basically two neighborhoods of radius 2. So what it means is that every point basically that I connect here to the central point is at most 2 links away. Okay. So you could look at basically radius 1 neighborhoods, radius 2, radius 3. It turns out in 2D, and that's an important point, basically 2 is a kind of optimum. And the reason is that if you just look at nearest neighbor, you get a very limited number of topological motifs. If you go to radius 3, you get a combinatorial explosion of the range of motifs. So computations become extremely difficult. So 2 is a sweet spot where you get, say, even a 2D tissue of the order of 1,000 different motifs. And what you can do then is basically count the relative frequency of these motifs. So, and in order to be able to do that, basically you need a way to label and really count each of these structures, maybe up to some sort of isomorphisms, basically. So now this can be done. So that goes back basically to work by Weinberg. So what he basically said, so you can pick basically any graph, you just have to make sure that it basically doesn't have any basically leaves just going off. And you can basically start at some vertex and you can traverse basically that local graph so that you at most hit each edge twice. So when you do that, basically you write down basically the sequence in which you have visited all edges. And then you start basically with a different edge. And you do that basically in all possible ways. And in the end, basically, you can lexicographically order all the basically names that you got. So you can think of each of these paths, basically sequences, as a name of one particular labeling. And you can order them basically so that, you know, you have most ones to the left, most basically ones to the second spot, and so on, and you go on and on. So you get a labeling, and you take that as your name. And what he showed is basically that this is a unique identifier, basically up to graph isomorphism. So it's a way basically of simply assigning in a unique way labels to these planar graphs. I will show later briefly a generalization that Dominic constructed to do it also for 3D structures. So where you basically have to look at simplicial complexes. Good. So basically what we have a labeling. So now basically we can ask another question. We can ask basically how similar or dissimilar are these two motifs here. I give you basically two motifs. I can ask, so how much effort would I have to make to basically change one into the other? So the way I would do that basically by taking basically the nuclei, moving them around. And the basic units basically that's well known is basically that you could basically look at topological D1 transitions. So I think most of you already know that.
So how big are the motifs?
So we take radius 2. Yeah. So for here, in 3D, we can take just nearest neighbors because there we have enough, basically a big enough space of different motifs so that we can characterize different structures. So it's really, it's a numerical argument. So the more basically, the bigger you make your radius, the more basically, the finer you can distinguish your systems, but the more computational complexity basically you accumulate.
Sorry,
Yeah.
this looks like in vertex mode contains something called a half-edge data structure to follow where of the node.
Yeah.
Similar, right?
Yeah. So up to here basically, so we use just things that are known. I just basically try to explain basically what we have. So we have basically different motifs. Basically, they all have radius 2. Basically, for the first half of the talk, they all have a unique name assigned to them. So we can look at them and basically just count them anywhere. But the important thing where we gain something is that we think about basically how far any two motifs are away from each other. And we think and basically how many elementary basically T1 transitions do I need to go from say this one to this one. So T1 transition simply means basically you have basically here say initially the blue cells in contact, and then basically something changes, and after obviously the transformation you have the yellow cells in contact. And so if you look at the triangulation of the midpoints of these cells, obviously you basically see that type of flip. Yeah, so that's called a flip. And in a second, basically, I will tell you basically why that is useful to have. So what we can do now is we can a priori, before we look at any specific system, create basically a graph where each node corresponds to a specific motif. Okay, so these graphs are simply basically our nodes, they're the motifs. And I connect them if they are one flip away from each other. So what that gives me, or what I gain, is that I have a metric basically structure now, a distance structure between motifs. And that's something that we will exploit to get better distinguishability of different patterns. So now what we can do for any given tissue, I can pick basically however many cells I want. I have to pick a large enough number. And I count basically the motifs, and I can think of that basically assigning a weight to each of these vertices. So I put a probability basically of finding basically a certain number of times this motif or a certain number of times this motif and so on. So every different system will be characterized by a different probability distribution on the same underlying flip graph. Yeah.
I guess there's a reason for the flip to give a good metric, right?
Yeah, I mean it's basically also an energetic unit in a way. So it's like it's a simple thing.
Simple thing that's not.
Yeah. So it's you could do more sophisticated stuff, but it turns out for our purposes this is good enough. So now basically if I think about two pieces of tissue, also the same tissue at different times, so it will have slightly different distributions because something will have changed. So now I can think about ways of comparing these two distributions. So what basically one could try to do is simply basically use entropic type measures. So Kubrick-Leibler type distances, you can symmetrize them to get a proper metric, so you use variation of information and so on. But what this does not use is that the underlying space basically has a metric structure. And it's known basically in other areas that if you have an underlying metric structure, you can basically gain something in terms of if you exploit that structure. And that's basically where optimal transport comes in. So the way we think about it is really, so let's assume basically I'm given basically this tissue A and I want to transform it into the tissue B, which means I have a probability distribution here and I want to transform it basically to say this probability distribution. Then there exists something which is called typically a Wasserstein metric or optimal transport distance, and you can define it in slightly different variations. which basically penalizes basically how much mass basically you transport over a given distance. So that's the idea here. So the way it works in practice, basically, if you put it in a naive way, is basically, so we have basically the flip graph distances, so the DIJs is the number of links that connect certain nodes. And what I'm looking for basically is a matrix here, which is positive, so it's basically a probability matrix or joint probability that has basically my two distributions as marginals. And basically, I optimize basically over these basically entries here so that I get the smallest possible number. So that's basically the optimal way of transporting basically mass from tissue A to tissue B. So that basically gives me, so this is basically the way you can set it up basically, but you can compute it more efficiently by mapping it basically on a basically diffusion type problem where you take your basically mass distributions basically as the difference of those as a source, and you essentially solve an basically flux problem. And that basically can be solved faster. So that's just an algorithmic detail. Basically, that's known also for quite some time, but I'll come back to that.
So, but yeah, this is sort of metric independent. So your D.
No, I mean the D is a metric. You know? It's a distance. It's a distance. So there I use that information.
So, so what happens if I rescale? Did a rescaling of my.
So I mean, if you scale everything by the same factor, nothing happens. So basically, you just get a global. So what you're interested in is basically the relative distance.
So relative distance is
exactly.
It can't depend on attributes. You're comparing D's. Dij with Djk.
Exactly. Yeah. So let's imagine. So for example, I didn't have a connection here in that example. So I would have to go basically along this way to transport. So because I only connect these things that are immediately next to each other by T1 transition, because I think the other parts basically transitions are extremely unlikely.
From the pictures, there are some cells which seem to be a little bit larger than the others. Is that optical?
No, but it's not.
Or is it?
I don't know. I mean, but it's not a problem as long as your sample is large enough. Because all you do is really, you just need enough data to sample these probability distributions. Okay. So if you have basically mistakes, tracking errors in your data, as long as your data set is large enough, that won't basically hurt the probability distribution. If you're limited by data, then you have to think more carefully.
So just try to see when I understand it right. So you have a imperfect pricing material by the probability distribution of the region, right? Not the actual spatial pattern.
No. So what I do is I map any given material onto a distribution on a graph of basic motives.
Yes, exactly.
And then I shift mark along this graph.
And the DIJ is the distance on the graph.
It's a distance on the graph. Yeah.
So just number of things.
It's a number of T1 transitions that you need to go there. Yeah, exactly.
Yeah, so because you look locally, you're missing potentially global effects.
Exactly. So it's not clear at all that this gives anything useful. So, but it's a way basically that uses more information than just using entropic comparisons. So the question is that useful or not? So what we quickly try. So we tried it basically on a couple of test cases first. So one thing you can imagine is that, say, you go from a monodispersed packing of spheres to a monodispersed packing of ellipsoids of a given aspect ratio. There are two ways, or there are probably many ways of going there, but there are at least two ways basically of going there. One is basically you mix basically more of these longer ones into the smaller ones, or you start basically changing the aspect ratio of all these. So these are two different protocols basically of getting to something basically that should be. So now what I can do is I can just take basically of any of these ellipse packings, basically take the centroids, run through the analysis, and every entry in that matrix basically is a pairwise distance between two packings. Say between this space and this space and this space, this space and so on. So they show up at different. So it's a basically positive symmetric matrix. It's just basically all these Wasserstein distances. So now basically what it means, I have now mapped my materials on a bunch of points that have distances. So now I can take these points and can try to embed them simply in something that we can imagine. So like an R2, like a plane. So this is what you get basically if you use basically an MDS embedding, which is really just to PCA principal component analysis, where you try to basically lay a plane through your data in a way that you basically capture maximal variance. Just that the distance here is not given by Euclidean distance in a high-dimensional space, but actually given by this distance matrix. Okay. So now what's encouraging here is that we see basically that the topological measure, which just basically looks at a given snapshot, basically time, is able basically to pick up these two pathways, distinguish them. So this is encouraging. If that had failed, basically the talk would never have happened. Good. And what the other thing that's interesting is, so basically in this case, I mean, it's also a sanity check. So you need basically just 2 dimension because you have these 2 different protocols basically to embed these points. So basically you don't gain anything if you went to a 3rd dimension and so on. So this is, okay. But this is a way basically in which I will construct now in general basically diagrams to compare different systems. So I will compute, I use this topological distance to compute distance matrices simply between various systems. And those can be anything. So that's what I would like to highlight. So I can take these things, I could compare them to any other Delaunay tessellation. So I could, in the end, I will actually compare basically a packing of stars in our neighborhood to bacteria and so on, just at the topological level. All we have to do is basically compute this pairwise distance matrix in this form, and then compare basically embedded in some low-dimensional space, and then basically analyze the structure. So what I do now is basically just tell you, present a few examples, and then I will talk about in the last part briefly about the 3D case, which has some technical basically challenges, but it's not too bad. So one thing you can ask is, so if I take an active particle simulation, so it's just basically overlapping particles have some overlap interaction that are repulsive, and they can change their orientation randomly. So if I gave you just a single snapshot from each simulation, could you extract anything basically from these? Basically just a single snapshot. So what we have here is basically simulations that were performed at different filling fraction and at different basically levels of activity. So for each filling fraction, basically we scan through different values of activity. We got one snapshot, we took the snapshot, basically plotted him, computed distance matrix. So now when I do the same embedding in 2 dimensions, so I see that I actually recover basically the 2 parameters that are varied basically along the principal axis. So just from a single snapshot basically of active systems, I could order them according to basically activity within basically assuming that this model is a reasonable description. So that was something I mean we find also encouraging. Yeah, so the second principal component basically correlates strongly with activity. So the way you do it is you take all these points, you put them into the plane basically according to the MBS embedding, and then you simply color each point basically by the activity that you had or by the filling fraction. And you see if you do by filling fraction, it's basically pretty much the first and the second one. So I think it's quite interesting that you get just these basically scans from looking at a single snapshot. So there's nothing basically used about dynamical. Yeah.
I think I'm also a little bit. The PCA is on what?
On the distance, so you take the distance matrix. So you have a bunch of
distance.
On the distance, yeah. Yeah, because you take a bunch of points that have some distances. These come basically from our procedure. You could also take an entropic distance, you could take whatever your distance is, but you have every system basically has a distance to another system. And then you embed it basically just in a plane. If you plot these variance plots and the time decay enough, or basically doesn't level up, then you would add basically a higher dimension. So until you satisfy basically with the variance capture. Okay. So now, how could you use that in something maybe more interesting? So we can apply to developmental data. So let's assume you perform an experiment, and in the experiment, basically you have to fix your organism at different times, and you would like to know basically how these different snapshots are ordered in time. Yeah, so it's sometimes the case, the more modern basically protocols often allow you basically to watch dynamics, but basically for systems where you don't know exactly where they are timed, you could do that. So we took data basically from Susan Eaton's lab, published a long time ago. So what they did, they looked at fly embryo wing, looked at a small subsection. So they basically give video data. So we can take that basically to test basically at different times, but we basically forget about when the points are taken in time for our test. So we just basically say, scramble all of these snapshots basically that were taken at different times, compare them according to that metric, and embed them. So when you do that, you find basically that you actually recover also basically the time direction. And you can do a little bit more, that's what Dominic did. So we can actually understand a little bit basically about basically the special curve along which these different developmental stages lie. So that's also a way, just by looking at basically simple snapshots of systems, basically we capture time ordering. So which may be useful or not. If you have a video, you don't need it. If you don't have videos, then maybe interesting. Yeah.
How small subset of the snapshot do you take? So we can
Yeah, so we have some analysis basically about the downsampling. So it's fairly robust, basically for the typical experimental basically sizes. It works, but the key is simply you have to approximate the distribution of motifs sufficiently well. So that's so there's Dominic basically in that paper in there's a downsampling analysis. Check that.
So because there's some kind of resolution in terms of your.
No, I mean, it's typically, it comes with incremental resolution. So if you took a larger area, so you have more motifs, you approximate the probability distribution of motifs better. But sometimes if you have very large cells and your microscope has a certain basically field of view, you can only, maybe look at 10 by 10 cells or so.
Yeah, because I mean, I presume that's going to have some implication on, for example, how well you order.
Exactly.
Yes, yes, yeah.
I mean, it's not clearly not perfect, but I still found it surprising so that you can just look at these Delaunay motifs and you get pretty much your time ordering back. And there's something interesting basically about this. So I'll talk at the very end very briefly about it. So because we have distances between different topological motifs, essentially we have what's called distance geometry available to us. So it's developed a long time ago by Menger and various others. You see, where you can define curvatures and all sorts of things, basically given just a point set with distances. So you can think about geodesics, you can ask, is there something interesting in these curves, basically, in that topological space? So does it tell you something about in which order transitions are performed or so? So there's also some interesting math, so that you start basically from these packings and then you end up with something basically that allows you to talk about geodesics basically in these motif spaces.
You just know the ordering, and you know the direction of time.
No, I don't. You don't know the direction of time. I mean,
So it's either way.
Yeah. But I guess
You go from 1 to 2, and then you go.
But I would expect, that an experimental roughly knows basically.
Also the time.
You're absolutely right.
That's not
Yeah. We are only up to a -1 multiplication by -1. So, okay. Good. So I won't talk too much about this. We also applied it to bacterial swarming. So there's a nice data set if you're interested in active matter and want to compare also particle simulation to experimental data. It was assembled mostly by Hanna Jekyll and Knut Drescher's lab. So this is basically a bacterial swarm, and the swarm expands over several hours, and they have a microscope basically that takes little video snippets basically, and it detects the edge of the swarm and goes back and forth and so on. So you get essentially along this line basically snapshots. And if you click actually on the data server on every pixel, you will see a small movie popping up. And the movie tells you in principle everything about the collective state for a limited time, basically in that small window. So, I mean, yeah, if you want to connect active matter information or simulations to also gene expression and so on, I think this is a nice data set to start from. And so what we did, and this is just something I want to post here as an open problem because we don't understand it, is just analyze basically these packings in the same way, basically everything. And you can do that and it all works basically. But then we said, oh, let's just look at something else. Let's ask, so how frequent are certain motif sizes? So just count the number of neighbors basically in your table. So it's basically something more basic. And then we compared it to various different distributions. And what we found, so there's a comparison also on the paper, basically with various other distributions, that for some reason tracy rhythm seems to work best, which is basically related to eigenvalues in random matrices, and maybe because these are intermediately coupled systems and so on. So we have no idea. I think this is an interesting question to understand why you would see something like that, or maybe it's in some other distribution that just mimics tracy rhythm extremely well, basically. Yeah.
Which type of GU?
Actually, I don't remember off the top of my head, but
If you look at the multiple defects, you can use the same paradigm for this. You can define the block pneumatic and what the defect.
Yeah. You could do various things. I mean, so just we just basically did that for fun, and I mean, so one thing that comes out of that you have basically a 2-parameter essentially distribution of these motif sizes, which you can also use to create another embedding of your because tracy rhythm is a 2-parameter distribution. So
this is still more on the central class.
Yeah. So now I will come to some,
Yeah.
talk about something.
Maybe my question,
Yeah.
so I was going to ask, I'm going to ask it now.
Hmm.
So how does this depend on the aspect ratio of the bacteria?
Yeah, I mean, you can distinguish. So it's a very good question. It leads me to my next thing. So have you seen my slides? Good. So the next thing we are wondering, okay, so we could do it in 2D, but 3D is more complicated. So your tessellation or you are now basically tetrads in a sense. So you have to find different ways of labeling them. But what we were interested in understanding is that if you take biofilms formed by different species, and so one of the differences is aspect ratio. In this case, there are also other chemical distances. So could you just somehow distinguish them also using similar methods? So if you give me basically just basically a micrograph or something like this from a snapshot from a biofilm, could I make a guess which type of bacterium that was? Basically, if I have measured basically some reference strain before, so that is the type of idea. So yeah, so that was what we have to generalize is, as I mentioned, so we have to go from triangulations to tessellations. Here's just an idea for how Knut's lab basically obtains basically these basically images, so it's basically confocal scanning. And then so you do image segmentation, and now more recently also they have some basically machine learning annotated things, so where you can basically recover cell centroids and also various other quantities, but we basically forget about most of those properties and only care about the centroid positions and the triangulations that arise from those. Good. So what's the challenge? Yeah, again, so basically pipeline is very similar. So I have to identify the neighborhood motive. The neighborhood motive is now a simplicial complex. So it's basically tetrahedrons basically two together. And I have to assign a unit topological identifier. So you can do it in this similar way as I labeled basically before the graphs, just that you have to actually keep track of basically what your basic units are. So you can't do basically just vertices, but you have to keep track with your tetrahedron. So you get basically names or Weinberg vectors that are basically lists, basically that characterize, specify a given tetrahedron. So, and then you can also order them, and so that's also described in the paper, and Dominic came out from it. So maybe we should call it the Skinner vector or matrix. I don't know. So I didn't have anything to do with it. So it was all him. I just told him basically try to label these. So, but now basically we have these different motifs, and we have to again create the flip graph, which can you do? It's computationally more challenging because basically on higher dimension you have more combinatorial possibilities, and you have then to count basically these vertex weights in the same way. Good. So otherwise the idea is very similar. So I have not just basically these motifs in 3D, I have the flip difference. So it's also, you need a fast algorithm basically to figure out what the T1 transitions do in 3D. So it's quite basically in the paper. And yeah, and then you can go and basically compare these type of structures. There's one thing basically that we realized is, so in 2D, basically this traditional optimal transport works extremely well. And you can also do it in 3D, but it becomes painfully slow just because your space is bigger. But what you can do is you can modify the metric slightly so that it becomes computationally fast, but still does better than entropic distances because you still keep basically some structure about the flip graph basically and how far these things are away. And what it essentially amounts to is you start basically from your topological Earth movers distance, and you can rephrase it basically into a slightly different optimization problem. And then you make a simplification where you essentially just solve basically for a flux of a diffusion equation, for a stationary flux of a diffusion equation. Here is basically 1-0. So just to give you an idea, basically, so how this does. So these are now the different bacteria species. So we had roughly 10 samples from each experiment, basically different experiments. You can see already when you plot the distance matrix, you have these blocks. So that means you pick something up there. If you embed them, basically, you see also that you can clearly distinguish, for example, Salmonella basically from E. coli, and you can kind of distinguish also Pseudomonas from Vibrio cholera just by looking at the topological information. And now coming back to your remark, so if you color this by the aspect ratio, which is the coloring here, so you see a trend again. So that basically aspect ratio determines the topological property of the packing, at least roughly along this kind of diagonal. Good. So yeah, so now this is the one about uncertainty quantification. Slide is just So you want to make sure that you not just limited by sample area. So what you can do is you can take motive distribution from two different basically pairs basically and just mix them up. And you can see what would be the difference that you would measure. And then you realize that really, so if you look at the topological distances, so they are always outliers. So you can trust basically that you really pick up some signal. It's not just basically some sampling error that gives you variation in the distances and so on. Good. Here's another slide just that really compares different embeddings. I'm not going to talk about this one, but that's really poorly. But so here is the diffusion distance I was talking about. This is an optimal transport distance computed. So these embeddings, they're always basically up to global busy operation, like flip of axis or rotation. So you can, but what you see here is that, so the diffusion distance optimal transport, they do about basically equally well. Actually, this does even a little bit better. And the Jensen basically Shannon, so that would be just a comparison based on entropies. So does significantly worse. So it cannot basically single out really some of these. Yeah, and it's just the extra piece of information that you get from the flip graph that helps you. So in that sense, optimal transport uses more information, so it gives you better separation between basically probabilistic systems than just the entropic measures that are blind to the metric structure of the underlying space. Good. So, and now just to wrap up a couple of other applications. So this was we took data from previously published papers on the zebrafish brain, where the authors had already basically marked basically different types of tissues basically in the brain. You can embed it. So what you find is that from a topological perspective, actually all these different tissues basically form basically a one-dimensional topological line. I don't know if that's interesting or not, but you could think about curvatures in that space and so on. So you can do the same for the zebrafish video that I showed at the very beginning. So here it's actually from a different source, so it's that paper. And you can see also that you also in 3D, you recover basically here the time ordering, and you see some interesting features basically in these curves. So one hope is that you can use, for example, just looking at the topology to define certain developmental events in your packing. So the other thing going forward that I won't talk about is you have these busy tools now available from distance geometry. So where basically this is just a formula that goes back to Menger, basically for the Menger curvature. If I give you basically some set of points and I take 3 basically points basically in that set, then I can define basically a curvature basically in that space, basically by computing areas of triangles and so on. There are some subtleties here with we have L1 basically that is basically actually a straight line, so it's not a curved line. If you have L1 distances, because the direct connection here basically would be as far as here. So there is some interesting math stuff which I don't know if it could be interesting one. So now comes basically the main thing that I promised. So it's my last slide. So we were interested in just taking all the systems for which we could get sufficiently basically finely resolved 3-dimensional data. So we simulated some systems, we took experimental data, and we computed basically, or if I say we again, Dominic computed the distance matrix, and then we looked at embeddings. And then something curious basically happened that if you look, is now the 3-dimensional embedding. So this is a projection basically, if you look at the most dominant axis, and this is basically the projection basically where you also see the third axis. So then all the full symbols here, they are living systems. And the gray shaded area is the convex hull of those. Okay, so all the empty symbols are physical systems that we could find or simulate or in some other way. And then if you look at basically these systems, then the only thing that really sits basically in that living island or cloud are these foams. So we also here there's actually star position, so I find it always. So here are the stars. So if you look just at this projection, you may think, oh, they are living, but actually, so here they are basically slightly on the edge, they're slightly outside, but they're pretty close. So what Dominic basically has set up is a website, is the Topological Atlas, and if you have data from simulations or from experiments, so we'd be happy to add it because we're just curious if can you find a non-living system that would mimic the packings of living systems, for example. And yeah, so this is what I wanted to present here. So it's just basically also that the results are robust. So what I've presented here is basically how you can characterize and compare multicellular structures basically from various systems using neighborhood motive distributions. So the important thing is that you use basically also distance information, not just do entropic basically measures, basically if you use topological stuff. And I'm not sure how that exactly relates to race work, but I think the one thing maybe that as you throw more information away as you do topological, basically you may have to include basically this distance information to get the separation again. So there may be some trade-off here. Yeah, we don't understand why basically in many systems, so there are a few examples in all these papers, we find these crazy rhythm distributions. You can use basically these low dimensional embeddings to just from snapshots. So that's what I want to emphasize. So basically order systems, basically, even if say you have lost basically your labeling of your data files, so you could use that to kind of figure out which one, how they were relatively close to each other. And then there's basically this interesting possibility to define basically the distance geometry on the topological motive space, which may be a nice mark, basically. Okay. Yeah, thank you. I have to I have to start with Ray because he was my favorite.
Ray.
Things that the main thing, like cities or beehives or.
So we looked at pubs, the distribution of pubs in England, and there was something interesting, but I don't remember it. Yeah, but there would be in the first part of the talk. So we started looking at that after the paper was out, but we actually looked at this. Yeah, no, it's an interesting question. So in principle, you could do compare anything. So we have been contacted by theoreticians from the hyper-uniform packing community. And it seems, for example, that this topological measure can pick up different levels of hyper-uniformity, which they find interesting. So I don't know how interesting that is really, but we are happy if somebody wants to use it.
Okay. Yeah.
So it seems that your measure will apply really nicely to a short time frame, but the cells don't divide or die.
I wouldn't say so. I mean, so all you need to do is really, you give me a snapshot of your system and say you have the nuclei tracked. And you can ask, could you distinguish basically, say, you have maybe cells for a certain phase that just rearrange basically, and you would get maybe a continuous change, but then at some point, and that may actually happen here in this example. So when you have more cell divisions, that your distribution space may change, and you may get these more singular events, or say sometimes cell division at some point. So, but it doesn't matter. I mean, as long as we have tracked cells. So what we would hope is just that that measure would pick up these events, because we think it's kind of the simple thing that maybe an experimentalist could provide. So just fix your system, send us basically the data at these different stages, and then we see, maybe we could say actually here there's something happening in that system because the topology of the packing changes, and this may be due to I don't know, fluidization, or it may be due to cell division. We can't tell you what it would be, but we could tell you where it would happen, hopefully.
Okay, Marco. No, please, we go back.
Brilliant talk, and I have very main question. Have you tried to look at adverse entropy with these the different arrangements of the particles?
I'm not sure. I mean, we looked at. I have to go back. I mean, so that is. But we can check that. So we tested various things, but I don't remember. So there's a whole battery of things. I don't. Yeah.
I guess.
Maybe the previous question. So in your meta graph, it shows a flip as the base between the blank, but for instance, for cell birth, you have a different assume basically change. So how would your analysis be dependent on the choice of the base?
So I mean, the way we set up the framework is blind to these events, because you would give me one snapshot of your tissue, and then you would give me a snapshot at a later time where you have the nuclei at different positions. So we couldn't tell whether, so then we would count motifs, let's say of radius 2 if you have planar tissue. So, and we couldn't distinguish in principle whether that motif has changed due to a cell division or because a cell has somehow migrated or changed its neighbors. But what we could say is that the overall distribution may systematically change in a different way when you have cell divisions, for example. Does that make sense?
But if you would have chosen them, basically, like your graph would be different.
Yeah, so I mean, you could probably think about more sophisticated ways. We haven't done it because there are other ways also of changing basically these. And then, but then what you would have to do, you have to think about how to weigh basically in your graph, because you want to have basically a common distance structure still, at least if you want to stay in that optimal transport. So you could, for example, say, okay, I can get from this motive to another motive, not just through a flip, but through another connection in my graph. But I would have to maybe weigh that in some way. So that could in principle be done, but we haven't done it.
Okay.
Was a good idea.
If there are people.
No.
So, Jorn, fantastic talk. There was a picture where you showed these 3D structures, and then there were these triangulations, and they, to me, resembled like crumpled paper. And that's not very surprising because, you know, there's what, I mean, I know a little bit, but there are others that know a lot more. That, you know, the crumpled statistic, the crumpled paper, you can draw these different figures and then look at edge lengths and vertices and statistics and those. Any idea whether this compares or
No, I mean, we haven't talked about it. That's an interesting idea. We could try to look at it. Yeah. So one thing I have to say, so for all the examples that we've shown here, we generally neglect surface. So we take basically of all these point clouds, experimental point clouds, the interior, okay? Because you have to think a little bit more about how to include surface, but actually DOM has set that up. So it should be possible because one of the limitations basically of what I showed to all the systems, basically they were 3D, so they had really a nice 3D structure. So you could basically forget about the surface and then you have a nice interior. But many of the biological systems, so there are, I mean, maybe 3, 4, 5 layers. So your boundary basically is actually a large part of your system. So yes, some things set up, so there is data, so that could be interesting to look at.
Okay. Oh, Silke, and then
in either the folds or the cells, you don't necessarily need to do a demo on or you have the actual edge of gravity. Is there a difference in what you find if you use a Voronoi filing compared to the actual edge?
No, I don't know. I mean, so what we went by is basically we wanted to have one framework where basically people give us tracking data, and then we push a button, and it spits out all these constructions. So if you have data, it would be interesting to look into it in 2D system. I think IGEMA's group in the Netherlands, so they check for certain tissues that there's actually a quite close match between the Voronoi tessellation and the cell boundaries, but I have no idea. I assume, I mean, definitely in some systems like, you know, neurons or so, it should be very different. So I don't know. I mean, yeah. That's it.
okay, so there was I think Ray or around.
Very interesting talk because it forces a change of perspective. When I think about say solids and liquids and exactics, I think that the classification is based on what's happening at large distance. Your classification is based on short distances.
Yeah.
I'm wondering if there is some way of taking the data that you have and coarse grain it and see whether any of this information remains.
It's an interesting question. I mean, what you would do from a graph perspective is basically, wouldn't say graph classification, but you would need a rule, say, to construct vertices into single ones, I think. I mean, I don't see how you would in that framework because you still need points basically that you can triangulate.
What happened if you just took your thing and then somehow coarse grade, so you just made bigger objects and then did a translation?
I don't know. These are all great ideas, but I mean, Dom left. And I'm useless, so I can no. But I mean, these are all interesting questions, and yeah. I mean, you're right. So it's the local information that we look at, and I have no idea how the coarse grading works. So it could be that maybe you go to something that is kind of universal, so that you can't distinguish in that space. So you could, for example, plot such a diagram for basically taking your original point cloud, you somehow coarse grain it, which means you have to combine in a spin lattice, I mean, you would renormalize by defining a new spin and then you kill some of the others. And then you could maybe see where that leads you in a topological space. But I wouldn't see right now necessarily what the best procedure would be to, because there's some arbitrariness in how you would coarse grain.
Okay, Ray?
What you said, it seems like an interesting question to me is if there's a physical or biological system where you could turn on and off the life aspect of it, so that in one regime it's say just drowning particles, quasi equilibrium, and then you can turn on activity and somehow see whether your idea about distinction between living and non-living.
Yeah, no, that's exactly, it's a very good question. And we so actually one of the referees asked us, so what we speculate in the paper is that this is due to some sort of growth memory, because that's the most obvious thing. So what we then did is we and they asked basically for some sort of, I mean, more support of that hypothesis. So what we did is we took one of these tissues that had grown, and then we randomly rearranged the cells while preserving the volume distribution in that way. And then we find basically that these points move out. So the hierarchical way in which these structures are created seems to be somehow imprinted in what we see.
It sounds to me like the word hierarchy is very important here. Go by accretion, all the systems use better, but it's always, it's not like these, but it's sequential.
Yeah, no, we think that is somehow the non-equilibrium. So if you just shake basically a bunch of right grains, or you get, so it's somehow you see that at the level of these neighborhood motives.
Okay.
There is a big distinction in biological tissue between animal and plant, also bacteria. But plant tissue typically are much more solid than liquid in the ground. Do you have any idea of what happens with plant? Is there a distinction? I don't see any.
So we have one plant here, just this one, I think.
Oh yeah.
So
I will
at that corner, and whatever these guys are. So the bacteria are here somewhere, and these are the seabird fish. So they are
quite the opposite.
Yeah. So another question, interesting, I mean, related to race, is the remark is what would happen as things die, for example, if they approach equilibrium? Do they move out of that island somehow, or
you can shock it like you get.
Also possibilities of distinguishing healthy tissue versus cancerous.
Exactly. So I mean, we were hoping that somehow the organoid community maybe would be interested in that. We haven't gotten a lot of excitement from there, so we were happy that the hyperuniform community, it's a small subset basically you want to use it. So Dom is basically running basically their simulations through that, and you can clearly see that you recover it.
But they're already doing it in topological data analysis, this type of differences.
Yeah. So either bottom left one.
That's the one that doesn't.
Yeah.
I think you have a comment.
Yeah, so if you have elongated outcome about the impact, come back to this organoid, from the center of mass, it's going to give you French neighbors.
Yeah.
Right? So you can go around this by dividing the rods and little bits of more or less, and then redo that things got stable. I'm pretty sure if you do this, you get more believable neighbors. I'd be curious to see whether you get any difference in further steps.
Yeah.
That would be the natural thing to do with elongated objects or bad objects or whatever. You have to take into account the local geometry to just get better neighbors using some kind of extended order.
No, I agree. I mean, so what we looked at, so the motivation here really came from, I mean, these typical disordered systems where it's hard to say how close are those. I mean, if I just, there's an active matter example here. If I just showed you these snapshots, I mean, we could even make them closer in filling fraction or so on. And I mean, it's hard for me to find a way of just saying, oh, this is different in terms of topology from the other. So this was really kind of the motivation just to get some way. And also because I wanted to learn about optimal transport.
Yeah, just the methodological pre-step in some sense, but these are more like topical.
Yeah.
Okay.
No, but you're right. I mean, also, I don't think this is useful if you have other nice order parameters and you know more about your system. So I mean, it also depends on what you want to keep, what you want to throw away, what is the level of granularity you're interested in. If you have really nice elongated cell, I mean, pneumatic order parameters or these type of things may be more interesting than looking at purely topological ones. It's one way of looking at things with your disorders.
I did you said something about optimal transport. I mean, you said something, I think, to talk about the dimensional dependence and how things were much harder in high dimension. Dynamics got much slower.
But that's just a number of states.
So it's just really enumeration that's the problem.
Yeah, just the flip graph.
Yeah. So there, but fundamentally, apart from that, there's no fundamental difference.
Yeah. Also, these ideas, I mean, they're not new. I mean, the way, so maybe some of the implementation, but basically we went to different communities and we thought, so how do they solve it? What are the properties that you want to keep? So in hindsight, we probably would have used the diffusion distance in the first paper already because it captures everything. It's way faster to compute. And it uses the same amount of information. So basically something about the probability distribution and the underlying metric space. Yeah. But it's, yeah, other than that, it's just a combinatorial problem. How you set up the optimization to get the distance.
Okay, well, last question, and then we will have coffee.
Yeah, so if you just look at the distribution of the various neighborhood types before calculating distance, this are really signature. You do some AI or something like that to learn about it. Just from that.
Maybe, yeah. I don't know. I mean, probably, yeah. We haven't done anything in a long time. Yeah.
Okay, I think we're all done. So thanks.